So now we will move to our next very exciting part of the program. So now we actually asked some of the trainees to present, and these are folks that have they got the traineeship the first batch, and I think some of them might have presented over the years, some might not have. So we couldn't get everyone to present, but I know everyone after today would have presented in the past year at some time, hopefully. So so the first one actually, I think these were. Let me see. So yeah, we set them up alphabetically. So so the first is Elise Corbin. So while she gets set up, what we also said, we actually wanted each advisor or one of the co-advisors to just kind of speak a little bit and say a few words about the trainee and what they're doing. So while Elise is setting up, I'll just give her. I'll give a little introduction. So Elise actually is a mechanical engineering student, mechanical science and engineering. She did her master's with Professor Bill King, who works on AFM and thermal scanning probes and things along the likes. And then I had been collaborating with Bill on various other projects, and knew about Elise's work and her interests. And she was very interested in moving sort of from a purely mechanical, non-biological kind of work to more biological or biomedical related work. So we thought that would be a great example of somebody who's interested, and then how this IGER traineeship can help move her into this area. So we had nominated her, and she got the traineeship, and she's been working on using, actually it's work that was in our group that we started some years ago, on using mass sensors, silicon-based mass sensors, to measure the mass of cells and try to explore the growth and dynamics of cells. This is work we did with actually collaboration with Jimmy and others a few years ago, published some papers. And then Elise is taking that all to the next level, and she will talk about that in her presentation. All right, anything else I should say? Or? Okay, good. That's okay, you can get that set up. So Elise actually, this is a mini dry run for her prelim, which is on Monday. So Monday she's taking actually her prelim, so timing was just perfect, even though here this is a little bit shorter version of a bit shorter than the one on Monday, hopefully. Then go ahead, Elise. You have to put this on.
So hi everyone, my name is Elise Corbin, and my co-advisors are Dr. Rashid Bashir and June Kong back there, and as well as Dr. King in the mechanical engineering department. And I'm going to be talking about the detection of mass, growth rate, stiffness of signal adherent cells using our silicon pedestal sensors. So a lot of my work is actually focused around cancer and some of its characteristic traits. So really, what is cancer? Well, cancer is the uncontrolled proliferation of abnormal cells in our body. However, abnormal cells are made continuously in the body. So what differentiates them? Well, a normal cell, when it's damaged or defective, has regulators in which can send the cell through apoptosis or cell suicide. Cancer, this is not the case. Cancer will evade apoptosis at any cost, as well as it can self-signal growth, as well as it has an insensitivity to anti-growth signals. Also, it has this limitless replicative potential. And finally, as cancer grows, it grows in the form of tumors and actually wants to metastasize through the body. So what we're really trying to do here is characterize the mass of an individual cell. And this is important because cell mechanics such as mass and stiffness really have profound implications in cell biology, as well as tissue engineering in cancer and many other disease research. More specifically, the dependence between mass and growth rate really can help us elucidate the underlying mechanism of the cell cycle progression. So then, why single cell analysis? Well, as you can see from this plot down here, what we can really get from up to this point, most people have done populations of cells, and we can really get misleading results from this, as we can see by bulk dynamics. Whereas the single cell, we could be missing stuff through these bulk analysis. So, how do we weigh a cell? Well, we have a very special experimental setup where we use a laser Doppler vibrometer system in conjunction with a feedback loop and a lock-in amplifier to ultimately measure a frequency shift. So the idea is we have one of our MEMS pedestal sensors where we actually measure the resonant frequency of an empty sensor, and then we go ahead and add a cell or any sort of mass to that, and then we'll remeasure resonance frequency, and it's that shift between the empty cell to the empty sensor to the sensor with the mass that we're actually looking for. So down in the lower this lower corner here, we have an SEM image of our in-house sort of sensor, and as you can see, it's a 4-beamed spring design with a 60 by 60 micron plate, and the idea is we want to capture a cell anywhere on that plate. And what's so unique about these devices is the fact that, unlike a traditional cantilever MEMS sensor, the mass uniformity, this is virtually insensitive to where the mass is placed. So as long as we can capture a cell anywhere on that pedestal, we'll virtually be reading the same mass. So here we have a video of an MCF7 cell growth. And MCF7 is a breast cancer line that was originated in the 1970s from a postmenopausal Caucasian woman. And this cell growth is over about an 18-hour period. And as you can see, we go from actually, you have to watch the video carefully, but we go from a single cell to actually, it goes through a cell division at around the 13th hour. So if we kind of freeze frame at this point, we can actually see, and I've highlighted here, where the cell goes from a single cell through the cell division. And at this hour 13, we have this dip in the mass. And it's this sort of missing mass event that we believe is due to the cell partially detaching from the pedestal sensor, which decreases its overall contact area and appears as this missing mass. However, right afterwards, when the cell readheres to the surface, it goes back to its normal growth profile. So the next thing that I'd like to talk about is really the role of mechanical properties of the cell on our mass measurement. So following my MCF7 growth, I then fixed the cells and remeasured their mass. And fixation being that I stopped all the chemical and biological processes in the cell, and I did that using a 4% paraformaldehyde solution, which has been shown to drastically increase the cell stiffness. So what we have here is the apparent mass both before and after fixation. And what we see here is that the slope is not equal to 1. So there's this discrepancy in our measurement. And so for comparison, I also ran a fibroblast cell line, and I did the same fixation experiment right before and right after fixation. And again, we see this discrepancy in the slope. So, and we actually, if we compare these slopes, we see that they're not quite equal. There's a discrepancy there, but they're not the same, which is implying or suggesting that the viscoelastic properties of these cells is actually different originally before the fixation. So that's really kind of only the beginning of what we want to do, is we want to actually decouple what's actually going on in this apparent mass that we're reading. So in order to do this, we have to really look more closely at our measurement as well as our measurement theory. So the basic theory behind our mass measurement is this simple harmonic oscillator model. And however, this really assumes a point mass, where this is really not the case, because the cell can and will oscillate out of phase with the platform due to its viscoelastic properties. So if we include that sort of this layer of complication to the model, we actually would end up with this sort of 2 degree of freedom system. And this has been previously looked at, and it's been shown that mass and stiffness really have a very strong correlation. So what we wanted to do is then kind of verify this model through using yet another model of making pseudo cells of polyethylene glycol diacrylate, which is a hydrogel, which we're able to actually tune the stiffness of these hydrogels to whatever we want. And we can do that by simply varying the concentration of our gel by diluting it with simply DI water. So the difficulty, though, with doing this measurement is actually how do we deposit these pseudo cells that we're creating onto our sensors? Because at this point, they're fully released sensors, so they're extremely delicate. So what we've opted to do is using electrohydrodynamic jetting in order to deposit these. For those of you that don't know anything about e-jetting, here we have kind of a schematic of what it's doing. And so we have a gold palladium nozzle that we actually charge up to really high electric fields. And when that electric field kind of reaches a critical point, it will actually jet a single droplet at a time onto our surface. So we can really control kind of the size and shape of these gels. And here you can see some bright field as well as confocal images of these gels deposited on the substrate. And we deposited gels of 5, 10, 15, 20, and up to 100% gels. So we used confocal imaging in order to kind of render our volumes of these gels. And by rendering these volumes, we're able to figure out what the actual mass is of these gels. So what we did is then compared the actual mass to our apparent mass measured by our LDV system. And what we've seen is that there's at the very low percent gels, we can simulate that change that we saw with our cells, where the slope is not quite 1. However, at the very high percent gels, such as the 100%, we get that nice 1-to-1 sort of correlation, which is telling us that our actual mass measured from our LDV system to the volumetric mass, they're exactly the same. So what's great about using these pseudocells is we're able to measure the characteristic kind of properties of these gels. And we did so using AFM techniques, where I performed simply force indentation curves. So I used a very soft cantilever probe, and I came in contact and pushed into the surface to a set force, and then I retracted. And we're able to use this sort of elastic deformation range to ultimately extract our Young's modulus, or our stiffness. So, and what we found is that the solution concentration is directly linear with our elastic modulus. So at our very low gel concentrations, we're at a very low stiffness. So at this point, I want to bring everything kind of back together again. So using our 2 degree of freedom model and my experimental data, I'm able to look at the agreement here. So what we have is a top view of a 3D plot up here, and the red dots here are my experimental data. And if we actually slice through on this like plane, we get these plots over here, where the dotted line is the modeled data, and the red dots are the experimental data. And we can, as you can see, we've got really good agreement here. So our model is showing that we're actually quite on the dot. So what I want to do is I actually want to, for my future work, is that I want to look more at the growth dynamics of the MCF7 as well as the MCF10A cell lines and kind of compare cancer versus normal cells and see the differences in their growth dynamics, if it's exponential or linear. And I also want to look at the degree of metastatic potential, and we'll be using the B16, which is a mouse melanoma cell line, and these have been passaged in such a way that we can, they've got ones that are with a low metastatic potential as well as a high metastatic potential. And I want to use these cell lines to do the growth, as well as do some more of the fixation, and just try to finish kind of this decoupling of the mass and stiffness phenomena that we see. And I would measure the mass change before and after the fixation, and then also verify it using AFM techniques as well. So with that, I'll end, but I've got a few people that I want to thank because I couldn't do this alone. Dr. Larry Millett, Robert Free, James Peacold, Dr. Patrick Fletcher, Vincent Chan, Brian Zorvell, as well as Mitchell Collins for all their help in the lab. That's it. Does anybody have any questions?
So what's known about the changes in stiffness during the cell cycle in normal and transformed or metastatic cells?
Well, that's what I'm hoping to find a little bit more about. I'm still very new to this, and so I'm still kind of learning.
So this hasn't ever been done before? Nobody's looked at stiffness in
The
cell
stiffness
division?
of the cell before
During cell
During
division?
the
or cell division.
And then in cells that are dividing because they're metastatic versus normal?
So as of right now, nobody has looked at these different cases yet. So I'm hoping, if my models are correct and everything, that we'll actually hopefully be able to understand a little bit more about this. I can't guarantee anything, but I'll do what I can.
I noticed you use Calvin Watt model to model the cell. Right?
Yes.
And my question is, will the cell, the geometry affect your measurement?
So the geometry plays a very of the cell plays actually a very large role in the model itself. So if you've got a very kind of tall and skinny cell, if you'd like to think about it, that's going to change where we've got these sort of where this peak sort of occurs. It could be shifted over a little bit more to the left or a little bit more to the right. So up here, this peak, and that's where the geometry really comes into play.
So you're vibrating the whole system
Yes.
at a certain frequency.
Yes.
What the frequency is?
About 60 kilohertz in fluid.
So can you just sweeping the whole frequencies and get a broadband response of the mechanical problem?
So you would have to actually hold one side one side of the thing constant in order to get the stiffness out of it. But for our experimental purposes, we actually lock into the phase instead of the frequency. So doing a frequency sweep is not as simple as it sounds.
The model system is not for elastic, right?
Right. There is.
Elastic, viscoelastic.
It is a yes, it's a viscoelastic model. And I had excluded this for time purposes, but I also did some creep measurements to determine the viscosity of these gels as well. So what I did was I actually applied an instantaneous load using AFM and let actually the material then creep. And we use this region here in order to fit our Kelvin-Voigt viscoelasticity model, and we're able to then extract what our damping is, and which all went back into what my final model was. Thanks, Jimmy.
I just have a question on the weight, the iron mass.
Okay.
Since you're measuring for 20 hours, aren't you considering the cell, whatever the cell is secreting out, like all the excess of the matrix and stuff as part of the weight?
So that would, if they're being well, I would assume that it's all going back into the fluid around it, which would change the dynamics between the pedestal and the surrounding fluid. We're actually working on including a perfusion sort of chamber, so we'll be removing that waste, but that's not functioning at this time. I'll just say
There are
that.
some matrix proteins probably in that.
Not really MCF7, not from the matrix. MCF10A is will, but not MCF7.
So one I guess this issue related to all this is that we certainly, if there's other groups that are using cell lines, that would be a good model to try, like cancer versus non-cancer, or drug resistance versus non-drug resistance. I think it would be nice to start using sort of similar cell lines for various experiments. So if you have any suggestions, let us know.
I think they're similar, different cell lines, but do you think about what you're putting them on? I mean, do you have any polyolacing? Do you have any matrigel or collagen?
Collagen.
What are they on?
I do functionalize the sensors with collagen before I seed them.
Do-
Which which
It'd be interesting to see if you, when you do the 10As, if you actually get the spheroid type. It's a special geometry that, and then it'll have a basal lamina or basement membrane that'll secrete, you can detect with antibodies, and it'll hollow out the interior, goes through apoptosis in the middle. So you might be able to understand something about the matrices you're using with the 10As that have been really well characterized.
yes, actually one of the challenges that I've had with the MCF7 is they are extremely kind of overactive, and they don't actually want to adhere to the collagen that I place on. So I'm not sure if there's a better solution there. But I've been discussing that with Larry and stuff, trying to figure out if there's something better to actually use.
Along the same question, do you have a picture of the fibroblast?
No, actually, I do not have a picture of fibroblast.
So
Not in this slide presentation.
Oh, yeah, it should look different. One's epithelial and one's different.
Well, what's the source of your fibroblasts? Because every tissue has some fibroblasts.
Well, these were I used 3T3 fibroblasts. These were given to me just so I could do this experiment. I actually don't know very much about those.
Just be aware the heart
Yeah.
has its own fibroblasts, the skin, the
Oh,
kidney, and that might be something you want to get closer to.
Okay.
You
The
would want to use
source.
the human, there's a human fibroblast you can use.
Okay.
A mammary fibroblast.
Okay.
If you're going to use them, MCF7 or
Yes.
10As.
Yeah, that would really bring everything together, wouldn't it?
Great. Okay, thank you.